I'd like to start off with a land acknowledgement that was created by the PCC 593 autumn course. So as we begin our gathering, we would like to respectfully acknowledge that our event today is taking place on occupied Coast Salish land, and that the University of Washington is on the homelands of the Duwamish people. We pay respect to Coast Salish elders, past and present, and extend that respect to their descendants and to all Indigenous people. To acknowledge this land is to recognize its longer history and our place in that history. It is to recognize these lands and waters and their significance for the peoples who lived and continue to live in this region, whose practices and spiritualities were and are tied to the land and the water, and whose lives continue to enrich and develop in relationship to the land, waters, and other inhabitants today. And with that, I'd like to introduce the speakers for today's seminar. So we have Cory Morin, who is a clinical assistant professor in the Department of Global Health and for the Center for Health and the Global Environment, CHanGE. He received his PhD from the University of Arizona in geography with a concentration on climate and environmental influences on the ecology of vectors and vector-borne diseases. Cory recently completed a NASA fellowship at Marshall Space Flight Center, focusing on applying data sets derived from remotely sent satellite data to health applications. His research uses data-driven process-based models to simulate mosquito population and virus transmission dynamics with the aim of identifying climate and meteorological conditions that facilitate epidemics. Cory's recent work has focused on Aedes transmitted pathogens, including dengue and Zika viruses, and incorporating weather and climate forecasts into models for disease prediction. Thank you for being here, Cory. We also have Joshua Lawler, a professor in the School of Environmental and Forest Sciences. He's an ecologist driven by applied conservation questions and their real-world applications, with climate change at the root. In particular, he's interested in how climate change can drive shifts in plant and animal distributions and the impacts those shifts have at both the species and the ecosystem level. He uses a combination of field experiments and statistical and simulation modeling techniques, and works with collaborators to design tools that conservation planners can use to assess the impacts that climate change will have on protected landscapes. Additionally, he studies how climate change affects not only plants and animals, but also people. In this case, through the loss of ecosystem services such as carbon storage, as well as the ways that human health, climate, and the environment are connected. Thank you for being here, Josh. And with that, I will pass it over to the first presenter today.
Thank you, Tori. All right, I'm going to share my screen. Let's see, hopefully you're looking at what looks like a title slide. All right. So I first want to start out with an apology for the fact that I'm going to have to run right after I talk. And I would love to stay for the discussion because I think it's going to be a really interesting discussion, but I'm unable to, and I apologize for that. All right, so what I'm going to talk about is I'm going to talk about some of my work and some other work that looks at climate-driven shifts in species distributions, how species are moving around in response to climate change and how they might move around. We've known for quite a while that species are shifting their distribution. Species are moving in ways that make sense when you think about climate change. This was some work that was done by the Audubon Society that looked at the wintering distributions of birds over a 40-year period. And they used data from what's called the Christmas bird count, when people go out around Christmas time and they count as many birds as they can. They took those data and then they used them to map the wintering distributions of 305 birds. And then they compared the distributions from the mid-60s to 2005. And they found that of those birds, about 58% shifted their distributions to the north. On average, they moved 35 miles to the north, and some of them moved over 100 miles. So birds moving, they didn't all move, and they didn't all move northward, but a lot of them moved in the direction you might expect given a change in climate. We found, or some researchers have found, similar patterns for lots of species. Some of the early work done was on butterflies moving up the west coast of the US, butterflies shifting northward in Europe. But we've also seen studies that look at marine species. And this one looked at a number of different fish stocks, different species of fish around North America. And if you look, I don't know if you could see it, but on this map, there's lots of little colored dots that have little arrows pointing in the direction that those fish stocks seem to be moving. Then there are these big black arrows that point in the general direction of all those species in that area. And what you'll notice is, unlike what I described with the birds, they don't all seem to be moving in one direction. They seem to be moving in lots of different directions. And one might say, well, they're not responding to climate change. But the researchers of this paper, Malin Pinsky and colleagues, found that they were moving in directions that would get them to colder water. Whether that was deeper water or colder currents, they were generally getting to places where there was colder water. There are other species that are moving in ways that we might expect given climate change, and not necessarily always latitudinally. But some work was done this is a picture of Yosemite Park, looking at mammals. And a group went out and resurveyed a whole bunch of elevational transects that were done by Joseph Grinnell at the turn of the century. And they looked across this elevational transect at for mammals. And they said, are we finding the same mammals that Grinnell found in these same places roughly 100 years ago? And what they found is many of the mammals have elevations. Across the x-axis, all those numbers, each bar represents a different mammal species. And then the length of the bar represents the range in elevation that the mammal was found at. The green portions of the bar are where the mammals may have expanded their ranges, where they were found in the recent resurvey, but they weren't found by Grinnell. And then the red portions of the bar are where Grinnell found the species, but the resurvey did not. And so you can think of the green ones as potentially expansions and the red ones as contractions of the range. And you see that about half of the species seem to be showing a change. And of those, most of those were showing a change you might expect given climate change. Although some of them you see expanding to lower elevations and some of them you see contracting at upper elevations. Most of them are showing the kind of shift you might expect given climate change. So what about trying to project how species might move around in a changing climate? So now I'm going to talk about some of the work that my lab has done over the last 15 years or so to try to anticipate how species might move in the face of climate change. And some of the work we started out doing involved fairly simple models at a fairly broad scale, where we looked at the climates that species exist in today and asked where those climates are likely to be in 50 years and 100 years. And we built maps like this that show areas that are in dark green that are not suitable climates today. They're not climates you find the species in today, but they're places in the future where those climates might be. The magenta areas are places that have climates that we see the species in today, but they're not projected to have those climates in the future. So they might be areas where the range contracts. And then there's a light green area where the species exist today and the climates are projected to be suitable in the future. So you can think of these areas as areas of expansion, contraction, and then areas that might be stable. We used maps like that. We did that. We built those projections for 3,000 different species, and we looked at projections across 30 different climate scenarios, and we came up with 90,000 maps that looked like this. We then 90,000 maps that look like the one I just showed you, like that. And then we overlaid those to get a map like this, which shows the amount of change you might expect in any given place, given a certain amount of climate change. And so you can see in this map, there are some places in the Western Hemisphere that may see over 90% change in the fauna. You also note that there are some places that see over 100% change, and that's just because of the way we calculated turnover or percent change. But on average, across the Western Hemisphere, places are likely to see a fair amount of change in the fauna. And what we looked at here were vertebrate species. I said about about 3,000 species. They were birds, mammals, and amphibians. I had a student, a master's student, Carrie Schloss, who asked, she wanted to ask whether the species would actually be able to get to all those places that they might need to go. So if those projections are right, they may need to shift their distributions to get to those suitable climates. Would they be able to move to get there? So she looked at mammals. She took the mammals in the study. She took about 500 of them. And she asked the question of whether they'd be able to move fast enough over that 100-year period to keep up with the shifting climates that were associated with their distributions. So she first calculated for each species how far they could actually go in 100 years. And that took a bit of work because not all species have dispersal estimates. And so she had to do some modeling to figure that out. And then she looked at, in any given place in that species range, would they be able to move fast enough to get to the closest suitable climate in 100 years? And she did that calculation for every place in the Western Hemisphere that the species existed today. And she did that for each of the species too. And she came up with a map like this, which told us what percentage of mammals in any given place would be able to go fast enough, move fast enough over 100 years to track their shifting climate. And what she found is that on average, about 8% of mammals in any given place would not be able to move fast enough. But you can see that in some places, it's up to 40% of the mammals might not be able to move fast enough to keep up with their shifting climates. So the next question we wanted to ask, using the same set of studies, was, well, how are they going to actually move to get to these places? Assuming they can get there, assuming they move far enough, how are they going to move across the landscape to get there? Knowing that the landscape is not a blank slate, but it's covered with all sorts of potential obstacles, including highways and agricultural fields and cities and things that weren't there the last time species had to make major movements across the continents to respond to changes in climate. So we set out to look at how they might move. And the map on the right there that you're looking at with the orange and gold is a map of the human footprint. And it's based on land cover, it's based on roads, it's based on population density and nighttime lighting. And it lets you know how much of to what degree the Earth is basically influenced by humans. So we used that map and said, taking that into consideration, how would species move from the magenta, the pinkish areas, to the green areas? And how would they move from the lighter green areas to the darker green areas? So how would they move into these areas of expansion? How might they move out of these areas of contraction? And doing that in a way that would avoid these areas that are heavily impacted by humans. So we did that, again, for this time, over 2,000 species, again, these mammals, birds, and amphibians. And we overlaid all those maps to get an idea of where there might be particular routes that species might take. We then interpret that data with a visualization, and this is what it ended up looking like. The little yellow streaks on this map are routes that amphibians might take to move in response to climate change. The magenta ones are routes that mammals might take, and then the blue ones are routes that you might find birds taking to move in the ways that they would need to track climate change. I'm now going to shift gears and focus down at a finer scale, because if you think of the species moving, shifting its range to address climate change, there's going to be parts of its range that it moves out of, and species probably aren't going to move out of them as much as their populations are going to die back, and we'll see a loss of populations in some places, and then we may see species move into other areas. I'm going to talk briefly about a study that recently came out in Science that compared the changes seen in the Mojave Desert between mammals and birds. And so again, this is a group involving some of the same people that went back and resurveyed a bunch of transects or a bunch of trapping sites that were done by Joseph Grinnell about 100 years ago, and said, are we finding the same mammals and birds at these sites as we did? And they found something fairly interesting in that the bird community had changed dramatically. The abundance of certain species of birds had dramatically changed, but the distribution of the community of mammals had not changed as much, and these are small mammals. And they discovered it was for behavioral reasons. Mammals will be able to adjust their behavior, have been able to adjust their behavior to withstand the changes that have occurred already, but the bird community apparently was not. In my lab group, we've done similar study, or we've done studies at finer scales like that to try to anticipate, again, to anticipate what we might see in terms of the species that are there, taking into account certain species of concern for the regions we're working in. So this is a picture of the John Day River in Oregon, where a student of mine, Aaron Roosh, looked at how changes in stream temperature might affect the fish species there. And he looked at 3 specific species, and he built a model to relate air temperature changes to stream temperature changes. And it's not a simple one-to-one relationship. As the air changes, the water will change. So he built a model. It was a network model that took into account where you are in the river. It took into account some of the environmental variables determining temperature in the river, in addition to projected changes in air temperature. And he then evaluated in the future how much habitat, thermal habitat, we might see for these 3 fish species. So one of the species he looked at was juvenile Chinook, and he found that under a moderate change, projected change in climate scenario, that the Chinook would lose about 34% of the length of its thermally suitable habitat in this one portion of the John Day River drainage. Under a higher emission scenario, it would lose 77% of its habitat. The next species he looked at was the steelhead, and he found that they'd be a little more resilient in only losing 11% of their thermal habitat, up to over 40% of their habitat at a higher emission scenario. And the last species he looked at was the bull trout. Now, the bull trout is a species, it's a real cold water fish, exists only in headwaters, in colder waters. And he found that it would lose a much larger portion of the small amount of habitat it had in the John Day. We give you one more example of study we conducted at a finer scale, and this was to look at what might happen to wolverine in a changing climate. And for this, we used a very different kind of model. So we used a population model, a spatially explicit population model, to look at how populations of wolverines might shift or might change across the Pacific Northwest. Wolverines rely on a number of things for habitat, but one of the most important things that's related to potential changes in climate is snowpack. So they rely on snowpack for denning in the winter. And so one of my postdocs, Chad Wilsey, built a population model for the wolverine. He used projected changes in vegetation and projected changes in snowpack to model how populations of wolverine might change over time. The map on the bottom right that you're looking at is a map of what the model says current day wolverine distribution might look like. And it's only hypothetical because wolverine don't exist in all of those little yellow and green patches down in the US there. But they're places that the model said some wolverine could potentially exist. The other 5 maps represent population trends in wolverine in those watersheds that are represented. Any of those watersheds that are purple are projected declines in wolverine populations, and any that are green are projected potential increases. And you notice a couple interesting things. One is the maps are generally purple, and the other is that there's consistency across the maps. There are a couple watersheds that come out as either being stable or potentially being able to support even increasing populations through time. And they're consistent across those maps. And a lot of that has to do with the fact that there's a driver that's is consistently a climate driver that's consistently projected across these different climate change scenarios. All right, so that was sort of a whirlwind view of some research coming out of our group and others that shows how species might be moving around in the face of climate change, how species composition in particular places might change. And then the exciting part, I think, would be a discussion about at least some of these things. And the things I have listed here are really why we might want to think of how species movements could affect health. And I know Cory is going to talk about the first one to some degree. But one can think of also ways that new invasive species could affect health, either by affecting crops or by affecting economics, which could then affect health. Pests and pathogens are moving around because those are species too, and they can affect the rate of forest fire, they can affect forest health, they can again affect economies, they can affect crops. And these things translate into effects on food resources and potentially on cultural resources, which can have an effect on health, whether it's certainly on physical health or on mental health. We can think you can think of potentially more ways that species moving around could impact health. All right, I'm going to stop there, or maybe there.
We could give Josh a hand clap with the reaction thing as he's going to have to say goodbye. Okay. Thank you, Josh.
Yeah, I apologize.
Okay. Cory.
Great, thanks. So I'm going to share my screen. So if it looks a little awkward, I have my PowerPoint on one computer and my video on another computer. It looks like I'm not looking at you, but it's just I'm looking at the other screen. Let's see. All right, can everyone see the screen now? Perfect, great. So actually, this is a pretty good pairing, going off kind of what Josh talked about in species movement, because I'm going to mention it a little bit, just because when you talk about infectious disease and climate, the impacts of climate change on the range of vectors, hosts, and then the pathogens themselves naturally comes up. So I'm going to go, most of my research has been on vector-borne diseases. I've done a little bit on some other things. We're actually starting to do some research on snake bites, actually, and how the number of snake bites or the seasonality of snake bites in some place like Arizona, where they do have a lot of rattlesnake bites, could occur too. So disease systems are, I think, very interesting because they do live at this kind of intersection between human systems and natural systems. And so there is this opportunity for people from very different disciplines to work together. And that's was somewhat helpful for me doing my graduate work in a geography department. We had people who studied things like climate, like I did, or geomorphology and biogeography. And you also had people who really studied governments and cultural issues and political issues. And sometimes they would work together. And one of the things that our project I was on was looking at West Nile virus. And from my perspective, it was interesting about how climate impacted the mosquitoes and the virus itself. While others were actually interested in the control problem and who has the right to, or whose responsibility is it to take care of mosquitoes? Is it private property that people should be doing it themselves? Does the county or local government have the right to go to certain places and make people turn over their buckets to get rid of the larvae or to spray? Obviously, spraying is a big issue where some people are concerned about the impacts of pesticides and so forth, and others think the risk of West Nile virus, some of the disease is much greater. But you get really these interesting connections and interesting challenges as well. And so when we talk about disease, there's something called the epidemiological triangle disease that some of you might be aware of. You can see it right here on the right where you have a host. Most of the diseases I'm concerned with are human, but there are obviously very important animal diseases as well. You need an infectious agent, something like a virus, but it could also be a bacteria or a parasite. And then of course the environment is important. And I'm focusing mostly on climate, but obviously there's other parts of the environment that are also important. And when you think about vectors, it also becomes more of a pyramid than a triangle, because you have vectors like a tick or a mosquito, which are impacted by both interactions with humans, usually because they're blood feeding, the infectious agent, and of course the environment themselves. most most vectors are things like our arthropods, which can regulate their body temperature. And so, of course, the ambient environment is very important for them. There's a few different categories of diseases that people should know about when they think about infectious diseases. You have human diseases, which are called anthraponosis, and these are diseases that are just generally transmitted among humans. And you have direct transmission, which an example would be something like measles, where the pathogen is generally just transmitted between the human population. Then you have indirect transmission, and this is when we start to talk about things like vector-borne diseases. And a good example of this would be something like malaria or dengue fever, which most of you have probably heard of, where the pathogen is transmitted from human to human, but it's generally principally transmitted through a vector. And in the case of malaria and dengue, it's a mosquito. So people generally aren't able to transmit the virus or the pathogen themselves. It has to go through that vector. And then you have zoonoses, which are diseases that are transmitted generally between animals, but once in a while will spill over into human populations. And so a good example of direct transmission for zoonoses would be something like rabies, where the rabies virus is transmitted between wild animals. However, very rarely a human can come into contact with an animal with rabies and it gets transmitted. But usually that's where the infection stops. Those humans don't generally retransmit to another human. And a good example of a zoonosis with indirect transmission would be something like West Nile virus, where the virus is generally transmitted between birds via mosquitoes. However, once in a while, after feeding on an infected mosquito, that mosquito can feed on a human and transmit the virus to the human. And humans are considered dead-end hosts. Same thing with things like horses and a lot of other animals, where once the virus is transmitted to the human can no longer retransmit the virus. So it kind of stays there, so they don't reinfect the mosquitoes. And that's something to consider when you think about kind of risk, right? If your neighbor has West Nile virus, you don't need to be worried about a mosquito biting them and then retransmitting it to you. The big risk there is that if they got infected with West Nile virus, it's likely that there's a pool of mosquitoes and birds in that area that actually are carrying West Nile virus. So I'll go into some of the reasons why climate is important for disease ecology. The first is how temperature can affect the pathogen. And there's something that I'll talk about a little bit later when I give an example of some study we're doing on dengue fever. But the extrinsic incubation period is a very important number, and it's really the period required for a pathogen to either replicate or develop in the vector before it's at a high enough level or at the right developmental stage where it can be retransmitted to another person. So this is important, that once a mosquito or other insect or other vector is infected with a pathogen, they can't normally just immediately retransmit it. It takes some time for the virus to either replicate if it's a virus or if it's a bacteria. And then for specific parasites, it takes time for the parasite to develop to the right developmental stage. And this is a good example of this is malaria. And if you look at this graph to the right, it's looking at two of the particular parasites that cause malaria, Plasmodium falciparum and Plasmodium vivax. It looks at the development time, right? The time for that once this pathogen gets inside the mosquito, how long it takes to go through all the stages necessary to be retransmitted. And it looks at that time at different temperatures. And you can see when you look at these very low temperatures around 15, 17, 19 degrees Celsius, the developmental time is 30 days or even longer. And so this is pretty important, right? Because most mosquitoes have a fairly short lifespan, usually 2 to 3 weeks. So if it's taking them over 30 days to, or it's taking the pathogen over 30 days to develop to the right developmental stage, it's very likely that that mosquito is going to die before the pathogen is going to reach that final stage. However, when you look at the higher temperatures around 30 degrees Celsius, then that development time is right around a week to 10 days. And that in that case, it's much more likely that mosquito will survive during that developmental phase, and they'll be able to retransmit the pathogen. So beyond just vector-borne diseases, just pathogen load and their ability to replicate in the environment, if it's a bacteria, for example, is oftentimes controlled by the ambient temperature. So generally, these bacteria or other pathogens have an ability to survive in the environment, but that ability to survive is generally dependent on what the temperature is. It might need to have very warm temperatures or cool temperatures and so forth. So temperature also affects the vector and the host. So when you think about mosquitoes and ticks and other insects, we generally think about those being kind of summertime organisms where, in the summertime, the mosquitoes come out, the insects come out, and you have to start worrying about them, but not in the wintertime. And this makes perfect sense. As I said, they can't internally regulate their temperatures. And so generally they either die off or they go into a hibernating state. And so you won't see them feeding. They might just kind of seek shelter and remain there for long periods of time. And then of course, this impacts their population dynamics. So they might not be developing very quickly, and their survival rates might decrease quite a bit. So the two graphs at the bottom are for Aedes aegypti mosquitoes, which I'll talk a little bit more about later, which transmit things like dengue fever, Zika virus, and some other important like chikungunya and some others as well. So the graph on the left shows the proportion of larvae surviving through their larval stage to their pupal stages. And again, you can see that at these very low temperatures, 8, 10, 12, up to maybe even like 15 degrees Celsius, they essentially die out. They're just not able to survive. And then it goes up quite quickly. They do best around kind of the high 20s, low 30 degrees Celsius. And then when it gets too warm, above, in this case, it looks around 35, 36 degrees Celsius, they tend to die off again. So that's an important thing to consider, is that people generally tend to think warmer temperature is better for mosquitoes. And that's true to a point, but at some point it actually gets to be quite detrimental to them. And the one on the right is, instead of survival, this is actually the development per hour. And so again, you can see is that temperatures increase, they tend to develop much quicker. And this kind of makes sense as well. If you have warmer temperatures and the temperatures increase the development rates, you're going to have bigger populations. Precipitation can also be important, especially for contaminating drinking water. So when you get lots of rain over the surface, it can actually pick up fecal material and other things and contaminate drinking water. It can also change the temperature and the chemistry of the water as well. I'm going to try to talk a little bit closer to my microphone. I think some people were having some audio issues. So yeah, precipitation can be quite important for the pathogen itself. And precipitation can be really important for vectors. So we think of things from mosquitoes, they have to go through a larval and a pupal stage, and they need to be in water doing it as well to complete those stages. So when you have precipitation, you tend to increase the amount of standing water, and that increases the amount of habitat the mosquitoes have for their reproductive cycle. But one thing that's interesting is too much rain can be detrimental as well. And so one thing they find is after hurricanes, you get a lot of rain and you tend to knock down the vector population by washing out all their breeding habitats. But then after that water settles down, you actually get a big explosion a few weeks later, because now you've had lots of standing water for the population to kind of to regrow. There's also interesting scenarios where in Florida, they found that actually low rainfall is very important, because when you have low rainfall, you tend to evaporate a lot of the water in the area, and you get these kind of very small standing pools of water. And birds and mosquitoes tend to congregate around these small water sources. And when they congregate around them, there's more contact between them, and this actually facilitates more transmission of the virus. And of course, increased stored water is really important. And there have actually been a couple of studies in Australia that show that their risk of dengue is often tied to people storing water. And so they're able to support Aedes aegypti mosquito populations in areas that generally would not be permitted. And so drought can also be a risk factor for certain vector-borne diseases. And of course, vectors also affect their host. So if you're a bird species, in the case of West Nile virus, what your behavior maybe might be determined by what the climate is like. So for example, when you have those drought conditions and the birds are looking for water, they might again tend to be in more contact with mosquitoes. Crows are especially susceptible to West Nile virus. And during the kind of big epidemic stage during, I think it was 2001 through 2003, where West Nile virus essentially swept across the United States, there were places where the corvid population, like the crows and jays, actually got very decimated by West Nile virus. And then of course, people outside when it's nice outside, during spring and summer, lots of people like to go hiking, and of course, you get lots of vector human contact there. When we looked at a study in in Arizona, the golf courses were important places for vector human contact, specifically because in a desert area, there tends to be not a lot of water, but kind of some of the golf courses, some of the locations where there were lots of water sources and lots of mosquitoes. Humidity can also be very important for both pathogen survival and vector survival. So the graph on the left is actually showing influenza activity at different levels of specific humidity. What's really interesting about this graph is how that is kind of horseshoe shaped. And so you see these low specific humidities and these very high specific humidities tend to be very favorable for influenza activity and transmission. This has ramifications for the seasonality and how it changes by location. And one thing they found during the study is that places in temperate regions tended to have high influenza activity during their winters because their winters tended to be rather dry in this area. And places in the tropics tended to have seasonality that was more related to their wet and dry season. And they saw their wet seasons where they got a very high specific humidity tended to be where they get most of their epidemics. And over here, we're looking at vector survival for Myxodes scapularis, which is one of the ticks responsible for Lyme disease. And so here again, you can see vector survival on the y-axis and the exposure time on the x-axis. We can see for short exposure times the different humidity levels, with the darker colors being higher humidity levels and the lighter being lower humidity levels. At those higher humidities, they're able to survive basically the same as the lower humidities. But as you go for longer exposure times down to 16 to 20 hours, you can see that survival decreases greatly when you have a much lower humidity. I'm going to show you another graph later for mosquitoes where this is also true. And it just happens to be true that most arthropods are not very good at maintaining their hydration. And so very dry environments, like if you're in an air-conditioned building, oftentimes the insects are not able to survive very well. Wind is not as well studied, but it can be important for things like soil-borne diseases, where if you get a lot of a pathogen in the soil, like valley fever, which is caused by a fungus that lives in the soil in the southwest part of the United States, like in Arizona and places in California. So when the soil gets disturbed and brought into the air, you get a lot of inhalation, and then you can get increase in disease. And of course, something we already talked about a little bit today that Josh talked about is kind of climate change impacts on species distribution, not just animal species, but he also mentioned diseases as well. And so on the left here is a graph looking at the reproductive values for Lyme disease for a few different scenarios across the eastern part of the northeastern part of the United States. And you can see that there's kind of an increase from the kind of present period, or base period, which is kind of 1971 to 2000, up until the 2040s to 2070s. So you can see that you see this big increase in areas where it's already present, and you see an expansion further into Canada. And this is something Canada's had to deal with, is increases in the range of ticks and the increase of Lyme disease. And on the right, this shows mosquito populations. The mosquito population is on the y-axis, and the x-axis is the week of the year. And then it has two graphs of the population, this dotted line, which represents a projected future scenario, and the solid black line, which is the present basis scenario. And again, what you can see is you actually see this dip during the summer because you get really warm temperatures that are actually a little bit more detrimental to the mosquito. But you also see an increase in the seasonality, where there's an expansion during the spring and during the fall and winter because of the warmer temperatures. And one important thing to consider is the scale at which all of these weather and climate phenomena impact disease ecology. So the first one we'll look at is temporal scale. And we think of like kind of a daily impact, and you think of things like a weather event, like I talked about a hurricane just wiping out a habitat for a mosquito, or a frost, which could really kill a vector population. And then you can think of a little bit longer scale. You can think of like weather systems that roll in over kind of like a week or two, and you can think of that being important for things like the incubation period, which we looked at for like Plasmodium, Falciparum, Plasmodium vivax, being around anywhere from like 1 week all the way to 4 weeks, depending on the temperatures. And then when you go on a further scale and you start thinking about seasonality, you think about what diseases diseases are important during what times of the year. And things like West Nile virus, for example, in Washington is not very important during the winter and spring. It's really kind of the summer and fall where it becomes important because the temperatures are and other conditions are right for transmission. And then going further, we talk about what Josh is talking about is really kind of the change in range expansion and so forth. And of course, spatial scale is also important. Obviously, climate and weather work on very different scales. So you can think of like mosquitoes, for example, a bush can be a very great environment for them if it shades them from really hot conditions. Or if, during a cooler time of the year, it's around a dwelling that's being heated, they will survive a little bit longer. And then, of course, we go all the way up to the continental scale again that we've already talked about. And Josh did a great job of talking about it, of how when you kind of change your climate conditions, you see these expansions in the range of vectors or other or pathogens as well. So I'm going to talk a little bit about vector-borne diseases. For those of you who are kind of not familiar with them, it's just kind of any pathogen transmitted by other animals, by humans or other animals, by arthropods. And you can see kind of where the burden is highest, and you can see it's generally around the tropics. And just happens to be that during the tropics, you have a longer period during the year and warmer temperatures where a lot of vectors can survive. And here we see some of the most important vector-borne diseases worldwide. This is daily adjusted life year. So this isn't necessarily deaths. This is just the kind of the total burden of disease, including deaths, but also just disability that might be caused by being sick for, either your entire lifetime or even for a short period of time. Now, if we look at this list, malaria is by far the most important from a mortality perspective. Malaria kills about 400,000 people a year. Unfortunately, most of them are children under the age of 5. As bad as that number is, when I first started studying this, the number for malaria was somewhere around 1 to 3 million. So we have made great gains with things like better treatment and providing bed nets and so forth. But malaria is still a huge risk in many areas. Again, here are some common ones. Vector-borne diseases account for about 17% of infectious diseases, according to WHO, and about 700,000 deaths. But of those 700,000 deaths, again, probably at least 400,000 of that is from malaria. So they make up the bulk of it. And of course, for vector-borne diseases, there's many different insects and other, not just insects, but arthropods that transmit them. So when we talk about mosquitoes, we have malaria, which are these Anopheles mosquitoes. West Nile virus is transmitted by Culex mosquitoes. And then Aedes aegypti are for dengue fever, Zika. It's a very prolific vector. It's this one right here. It's very easy to identify by these kind of black bands along its legs. Then you have flies. It's a black fly that causes onchocerciasis or river blindness. Leishmaniasis is a sandfly. Chagas disease are caused by kissing bugs, which are up in this top right corner here. And then ticks, here's a tick right here. So Scapularis, which I already mentioned, is responsible for Lyme disease. But there's also other important tick diseases like Rocky Mountain spotted fever. And these diseases, their ecologies are very different, which means their impacts from climate are also, weather and climate are also very different. So I do want to talk a little bit more about dengue and the importance of both temperature and humidity for its survival and impact on its range. So if you look at this graph to the right, the colored lines here are showing the survival rates at different temperatures and humidities. And so you can look at the red being very low humidity and the green being the higher humidities. And this shows you what we kind of showed earlier for the ticks, is that in general, the mosquito is able to survive at decent temperatures from -10 degrees up to around 30 degrees. But then when you get these extreme temperatures, they start to die off fairly quickly. But having that higher humidity tends to extend their survival. The red line showing around, maybe 36 degrees Celsius is when mortality tends to increase. So their survival is 0%. But when you get up to 90% humidity, it goes all the way up to around 40, maybe even 41 degrees Celsius. So that becomes very important. Another thing was when I talked about the EIP, the incubation period, and that actually decreases with temperature, and that tends to just kind of decrease indefinitely from, this is about 20 degrees, 20 days at about 20 degrees Celsius, all the way down to, like less than a week when you get up to these really high But realistically, it's going to be around here where you're going to get the sweet spot. We have a very low incubation period, but the survival still hasn't quite dropped. So if we look at this, we can actually calculate the probability that a mosquito will survive the incubation period at a particular temperature and humidity. And we did this for a bunch of municipalities in Brazil. In fact, it was 200 municipalities, all with populations about over 200 degrees. And we looked at, for each of the municipalities, whether they actually showed reported dengue cases or not. We came up with this right here, which was the probability of reporting a dengue case on the left axis for a, on the y-axis for a municipality, and the EIP, the incubation period survival probability that we calculated using their temperature and humidity. And this was done for each week of the year for 6 years for 200 municipalities. You can see, as you'd expect, that it increases, right? As you're able to survive the incubation period, the mosquitoes able to survive the incubation period, they're able to transmit dengue, and there's a higher probability of reporting dengue in a particular municipality. And we looked at the 50% level as saying, okay, that's about when we get about, it's about a little over 1% probability of survival. We actually really start to see a lot of dengue being reported. And we actually extrapolated that across the across not just the United States, but the North and South America. And what we have here is the number of weeks at each of these grid cells where the probability of dengue transmission or being reported is greater than 50%. Now, it's important to remember that this was done for using kind of Brazil as an example. And because of that, dengue is endemic in this country. So there's much easier risk of reintroducing the virus after it might have gone extinct. So even though you see, some probability, or you see 6 or 12 weeks up here where dengue could be reported, it's not actually going to be reported there, because this just means the conditions are suitable for dengue transmission. It doesn't necessarily mean it's going to be there. But again, what this shows is what you'd expect is that as you go latitudinally to the higher latitudes, you have fewer weeks during the year where you get transmission. But you do see areas, at least in the United States, like southern Florida, where it's pretty high. It's somewhere around, I'd say, around 40 weeks of the year. And this is one of the few locations in southern Florida and southern Texas where you actually do get local transmission of dengue in the United States. So if we know these kind of these conditions, we can actually start to use them to actually look at early warning systems. And this is really important because if you can detect, if you wait till you first detect a pathogen and then you respond to it, you're not able to do much damage to the epidemic curve. However, if you have a passive surveillance system, and you're actually able to have some kind of early warning system where you say, hey, at this point, we think that conditions are right for pathogen transmission, you can start doing some active surveillance, detect the pathogen earlier, have an early response, you can really knock down the epidemic curve. And this is important for doing preparation. This is kind of my idea of what you would do with this kind of knowledge and this kind of forecasting system, where instead of going through that normal cycle, you have some kind of environmental conditions that cause proliferation of a vector and virus and then cases of the disease. You can start doing some modeling early, identify the conditions earlier, and then you can start doing the process you would need to do in order to prepare for it, whether it's disseminating information to the public so they can take more protective measures, allocating resources for health authorities so that they can reduce the cases or at least reduce the burden of the disease, and then actually increasing surveillance. In that case, in the case of surveillance, you can actually use that data in order to feed your model and hopefully improve your model. So of course, there's some challenges with this kind of research, right? There's lots of uncertainty, both in the model uncertainty. You're also going to use weather forecasting for this, and there's weather forecasting uncertainty as well. And of course, your data is not perfect either. And so you also really need to incorporate both environmental and non-environmental risks. So most of my research is focused mostly on climate and weather, but there's all kinds of other important things as well. Socioeconomic conditions, what your healthcare systems is like, what kind of transmission intervention strategies are available. Those are all important. And there's still a lot of knowledge gaps, right? So the pathogen incubation periods are known for some of the pathogens, but not as well for others. For example, Zika virus is a relatively, it's actually not a new pathogen, but it hasn't been studied quite as well as something like malaria or dengue. So the impacts of temperature on its incubation period are not as well known. So with that, I'll end there and see if there's any questions. Thanks everyone for their attention.